Okay, our first speaker is Dr. Josh Leffler. He has BS in University of Utah, PhD in University of New Mexico, and his current position, he's a research assistant professor in wildland resources here at Utah State University. His areas of interest: plant physiological ecology, ecosystem ecology, and his current work is management of aspen regeneration in northern Utah and evaluation of management consequences to other components of aspen ecosystems. So, Josh Leffler.
Good afternoon, everyone. What I want to do today is tell you a little bit about a project that myself and several other members of the Department of Wildland Resources here at Utah State University are conducting with collaborators, with private landowners as collaborators at Deseret Land and Livestock here in northern Utah. This is a project that we're conducting to examine the effectiveness and ecological consequences of various commonly used treatment regimes that are used to restore aspen stands that are in decline. And I want to emphasize that this is a project that is just barely getting started over the last 2 years. So I'm going to be able to tell you a little bit about our rationale for doing this work, a little bit about the status of the project, the experimental design, the status of the project right now, and I'll finish up by showing you just a little bit of our pre-treatment data collection from last summer. But before I get started, I need to acknowledge many other people that have been involved in this work so far, particularly those that helped us collect field data last summer as part of our pre-treatment data collection. Without these folks, none of this really would have been possible. I need to acknowledge our collaborators at Deseret Land and Livestock. Lee, Steve, and Rick have been extremely helpful in getting this project off the ground. And then, of course, our funding agencies, which include USDA, NRI, Deseret Land and Livestock itself, and Rocky Mountain Research Station. Well, we're all here today at this conference because we all recognize the importance of aspen ecosystems to various stakeholders throughout the Intermountain West. Aspen ecosystems have a relatively lush understory, especially compared to surrounding ecosystems, and that understory is an important forage resource for livestock, especially in the late summer and early fall, when forage in valley bottoms is exhausted. For similar reasons, that lush understory is excellent habitat for game and non-game wildlife alike. Several studies have suggested that aspen ecosystems essentially represent a repository of biodiversity in the West. Aspen ecosystems are extremely diverse and harbor a disproportionate number of species compared to their extent on the landscape. That appears to be true for plants, birds, and insects. And aspen ecosystems are prized for their scenic beauty and recreation potential. But we're all also gathered here today because we're all concerned about the status of aspen ecosystems throughout the western U.S., particularly the extent of aspen on the landscape and potential loss of aspen over large areas of the Intermountain West. Charles Kay here at Utah State suggested about 10 years ago in a paper that aspen in the western U.S. may be doomed, a fairly dramatic prediction. Dale Bartos at Rocky Mountain Research Station here in Logan, in an analysis from 2001, suggested that possibly upwards of 60% of the aspen that was present prior to the arrival of Euro-Americans in the western U.S. may have been lost. And concerns about aspen are not necessarily new. Kronmiller, writing nearly 60 years ago, suggested of study sites in Northern California that there had been no surviving reproduction of aspen at that site for many years, suggesting that he also suggested that we may be approaching a crisis in aspen management. Well, concerns over aspen in the western U.S. generally fall into one or more of three categories. The first is conifer encroachment, and many aspen stands are early sterile in nature. In the absence of disturbance, conifers will establish in the understory of aspen. Eventually, in the absence of disturbance, those individuals will overtop aspen, and that stand, which was once aspen, will be replaced with conifers. Aspen dieback is a phenomenon that has been observed in many stands throughout the Intermountain West. Older individuals in stands have appeared to have died, and there appears to be no reproduction in the understory. And in many places, even where there is vigorous suckering, those suckers fail to survive due to the pressure of both native and non-native ungulates. The approach to aspen management in many cases is similar regardless of the state of the aspen stand, and that is partly because aspen is a species that will vegetatively reproduce when the canopy is disturbed. So management often revolves around disruption of mature canopy individuals with a hope of a vigorous suckering response. That vigorous suckering response will occur regardless of the treatment type that is used, as long as the aspen rootstock is relatively healthy. Various treatment scenarios have been used, including clear felling, root ripping, and even fire. But there's no reason to expect that all aspen stands are going to respond to management actions in a similar manner, because all aspen stands are not the same. For example, we might expect an aspen stand that has been heavily encroached by conifers to have a reduced suckering response than an aspen stand that only has a few conifer individuals present or no conifer individuals at all. Furthermore, different management actions may produce different levels of suckering response, and different management actions may have various consequences for other components of an aspen ecosystem. Specifically, some management actions are more intensive and therefore may be more damaging to other components in that ecosystem. We need to impose long-term monitoring plans on aspen management treatments if we're to examine their long-term effectiveness and their long-term consequences. We especially need to use long-term monitoring because we expect forests to recover from disturbances such as these management actions over multi-decade timescales. Furthermore, experiments are necessary to inform management decisions. Here, we are using a large-scale, landscape-level experimental framework so that we can develop management recommendations that will maximize the effectiveness of actions to restore aspen while minimizing the unintended ecological consequences. To that end, Utah State University has entered into an experimental agreement with Deseret Land and Livestock to examine several commonly used regeneration techniques for aspen in northern Utah. Specifically, we're going to be examining 3 different mechanical harvest techniques. The first will be a clear felling with removal of all of the timber from the site. The second will be a clear felling with that timber left in place on the site. And the third will be a selective harvest of merchantable timber. In addition to those mechanical treatments, we're also going to be examining fire as a treatment tool, and we'll be examining fire in the absence of mechanical treatment and with mechanical treatment previously done. But in addition to simply examining the effectiveness of various treatments in regenerating aspen, we're also going to be examining the ecological consequences for other components of those ecosystems. Specifically, we're going to be examining the effect of management actions on understory biomass and forage value, the species composition of the understory, the ability of the understory to serve as this repository of biodiversity, we're going to be examining the influence of browsing by both native and non-native ungulates on aspen regeneration in northern Utah. And we'll be examining the influence of management actions on soils, as well as the genetic composition of the stand. All of the work that we are conducting takes place on private land here in northern Utah, owned by Deseret Land and Livestock. Deseret Land and Livestock is a working ranch managed for cattle, sheep, cattle and sheep, as well as trophy hunting. Deseret is located northeast of Salt Lake City in the highlands of the Wasatch Mountains. In our experiment, we are placing 4 experimental blocks on the landscape, and each block has a similar arrangement to what I'll describe here. In our experimental blocks are first divided into 4 zones where we will apply our controls and our mechanical treatments. The first zone is our control treatment, where no mechanical harvest will take place. The next is our harvest all, or our clear felling and removal treatment, where all of the timber will be removed from site. The third is our selective harvest of merchantable timber, where we have a contract with a timber contractor that is taking what they deem merchantable timber from that site, and this includes aspen down to about 8 to 10 inches in diameter. And the last treatment unit is our fell-only treatment, where timber will simply be cut down and left in place. After the mechanical treatment is conducted, we'll be conducting a burn treatment over half of the experimental block. I should say that these blocks are about 400 meters by 200 meters in size, and the block is then divided into 8 treatment units, and those treatment units are each 100 meters by 100 meters in size, or about 1 hectare. After all of the treatments are conducted, we'll be erecting a fence over a portion of the control and the harvest all treatments, and this fence is going to be in place to exclude ungulates from a portion of those treatment units so that we can evaluate the effect of ungulate browsing on aspen regeneration. Operationally, how this is going to happen, or how it's happening right now, is that we're starting with our 4 treatment blocks, and we're first imposing a selective harvest on those treatment blocks, with the exception of the control area. After the selective harvest is done, the timber contractor is going back and removing the remaining trees from the harvest all treatment and felling the remaining trees in the fell only treatment. And after approximately 1 year, so about a year from now, we anticipate applying our burn treatment to half of the experimental block. We're allowing for a year in between the mechanical treatment and the burning treatment so that the green slash on the ground can cure and we have a chance of getting it to carry a fire. If all goes according to plan, at the end of next fall, we'll have our matrix of 8 individual treatment units in each treatment block, which will allow us to examine the efficacy and consequences of harvest by itself, burning by itself, as well as a combination of these harvest techniques followed by burning. So this project really got underway in the early part of summer 2007, and our first goals for 2007 was to select the stands that were where we would place these treatment blocks. We selected one stand and placed a block in one stand that was nearly pure aspen. We placed a block in nearly pure aspen so that we could examine the potential for regeneration in an aspen stand where we presumed that the rootstock was quite healthy. we placed 2 of our experimental blocks in mixed stands of aspen that had been heavily encroached by conifers, so that we could examine the reduction in suckering, examine for a reduction in suckering response when significantly fewer aspen were present in the ecosystem. And we placed 1 block in a conifer stand with only a few scattered individuals of aspen to ask if it's even possible to regenerate aspen in this situation. Our 4 blocks were all the position of our 4 blocks were all selected for uniformity in physiographic variables. All of our blocks are at an elevation of between 2,200 and 2,400 meters above sea level. They're all on relatively modest slopes of 10 to 30 degrees, and they're all on north-facing aspects. Our mechanical treatments began in the middle of June this year. As of right now, we're approximately halfway through the mechanical harvest of the blocks. Loggers are actually moving into the 3rd block tomorrow, and we anticipate that they're going at enough of a rate that they will be able to complete the mechanical harvest by the end of the fall. So hopefully, before snow flies, the mechanical harvest is done. The next goal for 2007 was to collect pre-treatment data in each of these stands so that we had some baseline data for comparison over time. And what we wanted to do was establish a data collection protocol that could be used before the treatment as well as for many years into the future. We based our data collection around a series of sample points in each treatment unit that we established along 5 transects through each treatment unit. Transects are spaced apart by 20 meters, and individual sample points on the transects are also spaced by 20 meters. Allowing for a 10-meter buffer around the edge of each treatment unit that yielded 5 sample points per transect, 25 sample points per treatment unit, or 800 sample points for the entire experiment. We used these sample points for all of our data collection last summer, and these sample points are all spatially explicit, so it's possible to interpolate data in between those sample points. The first data that we collected last summer was our stand assessment. We randomly selected 4 of those individual sample points in each treatment unit, and at those sample points, we conducted a variable radius plot so that we could calculate the basal area of each tree species present in the treatment unit. And we used a fixed radius plot for sampling the understory for regeneration of aspen individuals and conifer individuals. At each of our sample points, all 25 sample points, we examined understory species composition and understory biomass. We used a 0.25 meter by 0.25 meter nested frequency design to examine understory species composition. This is a nested frequency design, so individual species were scored as occurring in the smallest plot in which they occurred, and it's assumed that individuals that occur in a small plot are more frequent than individuals that occur in the larger plot. And after we sampled the understory species composition, we harvested our plot and used that plot to estimate biomass for forage value. We also, at 20 of the 25 points, we sampled soils to a depth of 15 centimeters. We measured litter depth, bulk density, and soil carbon and nitrogen content. And the genetic composition of the stand is being examined by Karen Mock in Wildland Resources. She conducted 2 different samplings of the genetics of the stand. One was a coarse scale analysis so that she can determine large scale changes in genetic diversity following treatment. But she also collected fine scale data so that she could examine the influence of herbivory on genetic diversity within a plot, within our stands, as well as selection by herbivores for certain genotypes in these aspen stands. So I'll use my last couple of minutes just to show you a little bit of our pre-treatment data collection. The first map that I'll show you is the basal area of aspen in all of our in our 4 treatment blocks. First thing I'll show you is that the block that we selected to be a pure aspen stand, all of the individual treatment units were greater than 80% aspen. The block that we selected to be our conifer stand, all the treatment units were less than 20% aspen. And the 2 mixed stands were quite a bit more variable. In one stand, the individual treatment units ranged from 20% to about 80% aspen content, and in the other, it spanned the full range from less than 20% to greater than 80% aspen content. In terms of conifer species, 2 of our blocks were dominated by white fir, and 2 of our blocks were dominated by subalpine fir. The other block, also present in the blocks were Douglas fir and Engelmann spruce. In terms of aspen regeneration, it's as you would have expected. Aspen, the blocks that had significant aspen present at the beginning also had significant aspen in the understory. Blocks that had relatively few aspen individuals had relatively few aspen suckers. Just a little bit of brief background, a little bit of brief data on our soils. We found that the bulk density of our aspen soils was considerably lower than our conifer or our mixed stands, possibly due to the presence of fossorial rodent activity in the aspen stands that's largely absent from the mixed or conifers. And in terms of carbon and nitrogen content of the aspen soils, the mixed stands were quite a bit lower in carbon and nitrogen content and had a much higher carbon-nitrogen ratio than either the aspen or the conifer. I'm not entirely confident of this conclusion because we've only analyzed a subsample of the data in the aspen soil, or in the soils at this point. In terms of biomass, we generally see a trend for increasing non-woody tissue and decreasing woody tissue as the basal area of aspen increases in the plots. And this is similar to this is as one would have expected from the literature. Final piece of data I'll show you because it is somewhat counterintuitive, somewhat contrary to what has been shown in other aspen stands, and that is that aspen is, in this case, we found fewer species in the aspen understory than in the conifer or the mixed understory. We've found about 47 species in our aspen understory, closer to 60 species in both the conifer and the mixed understory. So just to conclude, our mechanical treatments are ongoing, and we anticipate having those finished by the end of this fall. We're currently in the process of building our fence for livestock and native ungulate exclosure. We are right now clearing the path for the fence. Our burning treatments will take place in about a year from now, so in fall 2009, and we anticipate measuring our first response prior to those burning treatments in the summer of 2009, and we'll be measuring responses every 1 to 2 years over the next several years for this project. So hopefully with that I've given you a little bit of an idea of what this project is about, and I'd be happy to take any questions.
Why wouldn't your exposure go across all 4 treatment zones?
You want the truth? Money.
Not enough money for the fence?
We don't have enough. We don't have enough money to build that large fence over all 4 blocks. Initially we had hoped to do that.
Have you thought about constructing smaller fence in those other 2 blocks?
We have thought about doing that, and we may do that. Right now, one of the main reasons for the fence is the genetic sampling, and the scale of the sampling that you need to do for the genetic analysis requires that you have a relatively large fence. Otherwise, you could easily fence out genotypes, and you wouldn't see a response because you don't have replicate genotypes inside and outside. So that's the main reason for the decision about the size of the fence.
I'm interested in the issue that you found fewer species under the pure aspen stand. Are all of these aspen stands have been being grazed by livestock and wild animals?
All of the aspen stands are being grazed, certainly by both native and non-native ungulates. The grazing regimes at Deseret Land and Livestock are relatively minimal. We don't see a lot of grazing up in the high country. Cattle are not in there very frequently. In fact, I've been up there for 3 summers now, and we haven't had cattle in any of these basins. There have been sheep, but the sheep are only present for a span of 1 to 2 weeks.
Well, given Aspen's reputation of having high biodiversity, what do you account for than having lower vegetation species?
I don't have a really good idea at this point. We just got some of those data back, and I'm just as curious as you are as to why we've observed that. One thing that I haven't done yet is gone through and separate those data into individual functional groups. So if we take out just the forms, do we still see this response? And that I don't know at this point. Sam?
Do you think your plot sizes are big enough that you have a large number of genotypes, or are you looking at a couple of genotypes and your scaling may be limited?
Karen's sampling has encompassed the entire study block and areas outside of the study block as a single sampling unit. Our individual plots may not be big enough to examine a genetic response related to one individual treatment type. We're looking more for a larger scale genetic response to treatment in general. The plot size is partially chosen as kind of an operational constraint to fit these blocks onto the landscape, so that we could get as uniform as possible forest type on a single hillside.
As I understand, when you drew the different plots in the mix, the 2 mix, the different plots vary greatly between the percent of assets. So how are you going to be able to compare the results?
Probably more with a gradient type of analysis than a categorical analysis. It's probably going to be the best way that we go.
Okay, we have to move on to the next speaker. Thanks, Josh.